Tracy Quirk, and I'm an associate professor in the Department of Oceanography and Coastal Sciences, and we are the Wetland Plant Ecology Lab, and we're down here in the Mississippi River Delta, also known as the Bird's Foot Delta, in Delta National Wildlife Refuge. And we're doing a study to investigate factors influencing the die-off of a plant species called Phragmites australis, also known locally as rosocane or common reed. And it's been noticed that there's been some die-off of this particular plant, which is also a non-native plant. But it's an important wetland plant for this region because it's very good at stabilizing the shoreline and contributing organic matter to the soil. And so we have an experiment that we are conducting where we're evaluating how it's affected by flooding and therefore applicable to sea level rise. And these are some of the lab members that are out today helping.
Hi, I'm Sion Butler. I'm the research technician for this lab. I usually just help everyone with their research.
Hey, I'm Austin Lynn. I'm the postdoc in the lab, and I built these marsh organ structures to test how different levels of flooding affect the Phragmites or the rosocane. So we're out here checking how the plants are growing in the marsh organs. They're basically PVC pipe structures that we sunk into the marsh, and then we planted different varieties of Phragmites into the different elevations, so that some are not very flooded, they're like high and dry, and some are in the middle, and some are almost always flooded. So we're using this experiment to test how the percent time that they are flooded affects how well they perform.
I'm Olivia Hurley, and I'm an undergraduate lab worker, and I also do research for LSU Discover.
So we are preloading these syringes with some nitric acid, and that's because we're collecting pore water and we're interested in reduced iron. And so nitric acid tends to slow the oxidation rate. It's very hard to keep pore water anaerobic while you're in the field and transporting to the lab. So the best we can do is to treat it with a tiny bit of acid. And so we're preloading these syringes, and then we'll be out at the organs taking pore water.